Given the right temperature, pH, and nutrients, bacteria may reproduce as quickly as once every 10 minutes, as is the case for Clostridium perfringens, a common cause of food poisoning. It's important to know something about how microbes grow so that we can predict the effects of environmental changes on growth. Microbial growth is defined as an increase in the number of microbial cells. We can measure this growth by either looking at individual cells or the physical effects of large masses of cells. In this video, we will describe how bacteria grow and then look at how this growth is measured. There are three primary objectives for this video. The first is to briefly describe what happens during binary fission and to describe the terms generation time and number of generations. You also need to be able to calculate the generation time and number of generations based upon data that you're given. And then you also need to describe the phases of bacterial growth curves. There are many ways that prokaryotes can reproduce. Bacteria primarily use a process called binary fission. And as you might guess from the name, binary means two and fission means to split. So bacteria are going to split in two. Binary fission is a form of asexual reproduction. That means that only one cell is involved. In this case, the one cell begins to elongate as it reproduces the chemical components inside of the cell. So that means we're going to get two copies of the bacterial chromosome, that large circular bit of essential DNA. We might have a reproduction of the plasmids. Your ribosomes may begin to reproduce. And eventually what begins to happen is that the plasma membrane starts to fold into the middle of the cell. This is called invagination. Since the proteins that make peptidoglycan are embedded in the plasma membrane, that means that peptidoglycan gets formed here as well. Eventually, the two folds of the plasma membrane fuse together in the middle of the cell, and peptidoglycan is formed in that space. This produces a septum, or a partition, that separates the cytoplasm of the cell to make two new cells containing one copy of the bacterial chromosome. Eventually, enzymes called autolysins, which are in the area just outside of the plasma membrane, they begin to break down the cell wall. And this separates our one cell into two. The septum that I mentioned here in this slide, or in this figure, is also sometimes called a cross wall, by the way. Okay, a cross wall. Now would be a good time to pause this video and go watch the two videos in your lecture videos folder on binary fission. One of them is just titled Binary Fission. The second is a fluorescence study of binary fission. As a result of binary fission, one cell divides to give us two cells. And when those two cells divide, we get 4 cells. And when those 4 cells divide, we get 8 cells, because each cell is dividing to give us 2. 2 times 4 is 8. When these 8 cells divide, we get 16. When these 16 cells divide, we get 32. 32 cells become 64 cells. 64 becomes 128, 128 becomes 256, and so on and so forth. Each time the cells divide, we call it a generation. So the first generation is when that one cell became 2. And then the second generation was when the 2 cells became 4. Then the third generation was when the 4 became 8, and so on and so forth. I think you get the point. In microbiology, we actually use a shorthand to describe this. The shorthand is that we're going to take the number of cells that we get after a division, so that's the number 2, and we're going to raise that to the number of divisions, that is the number of generations. Assuming that we start with one cell, that means that after one division, so 1 times 2 to the 1, we're going to get 2. Okay, if we started with one cell and we had 2 divisions or 2 generations like this, then that means we should get 4. I hope you understand what I'm saying. So the simple shorthand is to put the number of cells that we started with. So far, we've only talked about starting with one cell, and then multiplying that times 2 raised to the number of generations. So that looks a little bit like this. Okay, so here are the numbers of generations on the left, and then you can see those as exponents here in the middle. And then the answer is over here. Now, as you can see, the number of cells that we get in just 20 generations is over 1 million. So the numbers of cells increases rapidly. We call this type of growth exponential. And I don't know if you're like me, but sometimes when I'm in a hurry to get something done, I make mistakes. So as you can probably guess, when you get to numbers that are this large and larger, it becomes very easy to transpose numbers or to drop numbers, and that just messes up your calculations and experimental results. So scientists have actually come up with an easier way of writing the numbers. This makes the numbers more manageable, and that's to take the actual number of cells that we've calculated here in this middle column, these numbers, take the actual number of cells and take the log of that. So we would say take the log of 32, for example. And then if you put that in your calculator, you should get 1.51. If you put the log of 524,288 in your calculator, you should get 5.72. So that's kind of how that works. Let's try a few practice problems. But first, let me review how to do exponents with your calculator. I recommend that you use your scientific calculator that you probably have from your chemistry class rather than your cell phone, but if you don't have that calculator handy, you can try your cell phone. In order to do exponents, you're going to look for one of two buttons. Your button may look like this, that's called a caret, or it may look like this. If you have a caret, like is highlighted in the figure on the left, then you could say to push the caret button and then whatever your exponent is, whatever the number of generations is. So you could put 2 caret and then 5 to mean 2 to the 5th. Okay. If you're using a calculator like the one on the right where you have the y raised to the x power type of button, then you're going to have to do a little test to figure out what's going on. It's a real simple test. If you have this button, put the number 2 in your calculator, press the y to the x power button, and then put in a 3. If you get the number 8 as your answer, then that suggests that the calculator is interpreting the 2 as your base number and the 3 as your exponent, or the y. If you get the number 9 as your result, then that means that your calculator is interpreting the 3 as the base, that would be the y, and the 2 as the x. So unless you do this test, there's not really a way to really know. So just take a few minutes and do that really quickly. If you have the calculator on the left, you can do the same thing just to make sure that you know how to get the exponents in your calculator and get the right answer. We're looking for the number 8 here. Okay, we're looking for the number 8. So 2 caret 3 should give you 8. Now we're ready to start with the practice problem. Here it is. A culture starts with 100 cells, and they are allowed to reproduce for 50 generations. How many cells will be produced? pause the video and think about that equation I showed you a couple of slides ago. You're going to need that to solve this problem. All right, let's take a look at how to do this. Remember, the equation that I gave you was the number of cells, which I'm calling X here, is equal to the number that we started with. Well, the problem tells us that we started with 100 cells, so we put 100 there. The next part was 2 raised to the number of generations. So you're told in the problem that there were 50 generations. So you should have an equation that looks like this. X equals 100 times 2 to the 50. If you put this in your calculator, you should get 1.13 times 10 to the 17 cells. So just a quick recommendation, you may want to put, if you didn't get that, you may want to put the number with the exponent in your calculator first, and then multiply the answer for that part of the equation times this part, times the 100, and you should get this number. Let's try another one. 10,000 cells were inoculated into growth medium, and they reproduced 20 times. How many cells were produced? You may want to pause the video, work the problem out on your own, and then press play to confirm your answer. All right, here we go. So here's our total number of cells, which is going to be X, is equal to the number that we started with. So the problem tells us that we started with 10,000 cells, so that goes here. Then we're going to say times 2 to the number of generations. You're told that the cells reproduced 20 times, so that's another way of saying there were 20 generations. So you put your 20 as an exponent. If you put this in your calculator, you should have gotten something like 1.0486 times 10 to the 10 cells. Now your number may not be exactly like mine, depending upon how you decided to round your numbers, but it should be somewhere in the ballpark. This type of question on an exam would be a multiple choice type question to help you be sure that you're doing the problem right and to eliminate getting the problem wrong because your number is different than mine due to rounding. Growth curves allow scientists to monitor bacterial growth over time. If we graph the number of generations versus the raw number of cells, we get the solid curve that you see here. There's nothing wrong with this curve, except that the numbers of cells are so large by the time we get to 20 generations or so, that we don't see the growth that's happening in the early generations down here from generations 1 to 5. The curve is actually suppressed, so it looks like there's no growth until generation 10 or so down here. And we know that's not true. Now, if we instead take the log of the raw number of cells, we get more manageable numbers, true. We saw that on the slide a couple of slides ago. But also, if we graph those numbers, our curve looks like the dotted line here. Notice that the growth rate, that is the slope of the curve, is pretty much the same at each time point. So we can see the growth rate in the early generations down here around generation 5, as well as the growth rate in the later generations, like around generation 20. From the slope of this curve, it looks like the growth rate was the same as we would expect. Generation time is the amount of time it takes for one cell to divide and become 2. In the introduction to this video, I mentioned how Clostridium perfringens doubles in about 10 minutes. Those 10 minutes would be the generation time. If we know something about how many cells there were in the beginning of an experiment, and we know how many cells there were at the end of an experiment, and we know the duration of the experiment, we can calculate generation time. Let me give you an example. If 100 cells are growing for 5 hours and they produce 1,720,320 cells, we can use the equations that you see here to calculate generation time. The first bit of information that we need to know is how many generations there were, right? Then we can take the time, which is 5 hours, and divide it by the number of generations. To figure out how many generations there were, we would take the log of the number of cells at the end of the experiment. So that's going to be the log of that 1,720,320 cells. So 1,720,320. Okay, and then we're going to subtract from that the log of the number of cells that we started with, which you're told that's 100. So the log of 100 goes here. After we subtract those two numbers, we're going to divide by 0.301. And this number, the 0.301, will always be the same. It's tied to that original equation that we were using where we were calculating the number of cells if we knew the number of generations. Okay, so if you back calculate, you'll get this 0.301. So that's the first part. If we do this equation successfully, we'll get the number of generations. To figure out the generation time, we're simply going to multiply 60 minutes per hour times the number of hours, which in your example here is 5. So we're going to have 5 hours. And then we're going to divide that by the number of generations. You should get something like 21 minutes per generation as your answer. I'm going to pause this video, or rather you pause the video, and try working this out. See if you get a number that's similar to 21 minutes per generation. Then join me on the next slide and I'll explain the numbers that you should have gotten and we can compare. Okay, so here's the problem. Here's the solution. So the first thing we had to do was figure out the number of generations. We took the log of 1,720,320 and subtracted the log of 100, then divided by 0.301. So depending upon your calculator, you can either put these numbers in first, so the 1,720,320, you put that in first and then press log, or if your calculator is like mine, you have to press log first and then put your number in. If you did that, you should have gotten a number that was around 6.24. Then you cleared your calculator and did the same thing for 100, and you should have gotten 2. If you subtract those two numbers together, you should get 4.24? And then divide that by 0.301 and you should have gotten about 14 generations. Then you could get the generation time by converting the number of hours into minutes. So we had 5 hours of time, right? That's what was here in the problem. Times 60 minutes per hour. Your hours will cancel out, leaving you with minutes, and then divide by 14.1 generations, and you should get 21.3, somewhere in there, minutes per generation. Let's try another one. If 32 cells growing for 24 hours produced 50,001,670 cells, what is the generation time? Pause the video, try this in your calculator, and press play when you're ready to see if you're right. All right, did you get 69.9 minutes per generation? Let's see how to get that. You're going to put 50,001,670 into the equation. So remember, it was the log times the end number of cells minus the log times the beginning number of cells. So we've got the log of 32. Then divide that by 0.301. So when I put the log of 50,001,670 in my calculator, I get 70.70. When I put the log of 32 in my calculator, I get 1.51. So I subtracted those two numbers and divided by 0.301. That gave me 20.6 generations. To calculate the generation time, I put 24 hours in my equation times 60 minutes per hour. Remember, this is just going to convert hours into minutes. So these two will cancel out. Then I divided by the number of generations to get 69.9 minutes per generation. So that means that these cells reproduce in a little over an hour. If you'd like more practice with the calculations around bacterial growth, there's a homework, an optional homework that you can do. So when we have a culture in the laboratory, it's not uncommon for researchers to monitor the growth of bacteria over a specific amount of time. Sometimes we do it until basically the culture is dead. If we plot the log of the number of cells, that's on the y-axis here, by time on the x-axis, we can get a curve that looks something like what you see here. There are four basic phases of growth in a bacterial culture. First, you have what's called the lag phase. When you add bacteria to culture media, they don't actually grow initially. Rather, they spend a bit of time assessing what the nutrients are in the environment, assessing the pH, things like that, and they make adjustments to their metabolism accordingly. So, for example, if you were to take bacteria and put them in milk, which has lactose, but the cells are usually metabolizing glucose, they may have to adjust their chemicals, the enzymes inside of the cell, so that they can break down the lactose. So during the lag period, that's what they're doing. They're changing their gene expression. After everything is all set and they're ready for growth, they will start growing and undergoing binary fission. They'll have an initial period of growth called the log phase, where the growth is exponential. The cells are highly metabolic at this time. They are rapidly undergoing binary fission. They are replicating their DNA. They are making lots of peptidoglycan, lots of ribosomes in cytoplasm, and they're splitting themselves in half. Eventually, because of all of the metabolism that's happening, you get an accumulation of waste products. What comes in has to come out, just comes out in a different form. So as those waste products accumulate in the culture medium and the number of nutrients decreases because of how many cells there are absorbing nutrients, you enter what's called the stationary phase. So during the stationary phase, the death of the bacteria is roughly equal to the number of cells that are being produced. So they kind of cancel each other out and the growth curve flattens out. It plateaus as a result. Eventually, the number of nutrients really starts to decrease and the number of waste really goes up. So the cells stop growing so rapidly. And the number of cells that are dying actually increases. So what we'll see is almost exponential death rate, where the cells will start dying more so than growing. So those are your phases of growth. You have the lag phase, the log phase, the stationary phase where growth is basically plateauing, and then the death phase where the number of cells dying is faster than the number of cells that are being produced. Quick question for you. Back in chapter 4, we learned that penicillin targets peptidoglycan synthesis. It weakens the cell wall and makes cells susceptible to osmotic lysis. Which of these growth phases do you think penicillin is going to be most effective? The log phase, right? Because there's lots of peptidoglycan synthesis going on as cells are undergoing binary fission and making new cells. We also talked about endospores back in chapter 4. Remember, those are resting cells that get made when essential nutrients aren't as readily available. Which of these phases do you expect endospores to be made? This one's a little more tricky. You might have said the death phase, but actually by the time the cells get to the death phase, it's too late. Endospores are usually made during the stationary phase. During binary fission, one prokaryotic cell becomes two. It does this by reproducing the components inside of the plasma membrane and elongating. The plasma membrane begins to fold in gradually, and a layer of peptidoglycan called the cross wall or septum gets built. This splits the cell into two. Binary fission, or the amount of time rather, that a cell needs to complete binary fission is called the generation time. And each time that a cell divides by binary fission, that's called the number of generations. In this video, we looked at how to calculate the generation time and the number of generations and the total number of cells produced after a certain number of division events. If you need practice on this, remember there's an optional homework in the folder for this lecture. The last thing we talked about was the phases of bacterial growth. Remember there are 4 of them: the lag phase, the log phase, the stationary phase, and the death phase. You'll want to know these for your exam, as there are usually a few questions about what happens during each of these phases. If you have any questions about this material, please don't hesitate to reach out to me by email or text. I'm more than happy to clarify things if necessary. Otherwise, I'll see you at the next video where we'll look at how a scientist actually counts cells to generate the data that we talked about in this video. I'll see you then.